flow cytometer. For us, when we are doing marine seawater samples, we use it to look at bacteria, virus, Chinchococcus, cyanobacteria, and Prochlorococcus. Okay, bacteria and virus you have to stain, but for Chinchococcus and Prochlorococcus you don't have to stain, but they have different wavelengths, different colors, and they are of the picoplankton fraction. Picoplankton fraction means they are less than 2 micron in size. So before you can start your analysis, you must of course run through your calibration beads. Okay, so we had 2 micron and 3 micron calibration beads. So we ran through the machine and we obtained the following dot plot. Okay, so for this flowing software, what you do first is you have to create the visualizing tool. Let's just say you create your dot plot, and then only then you open up your file. Okay, you open up your FCS file, Flowcyto file. Okay, so this is the standard file. Okay, you don't see anything here because the channel is wrong. So what you do now is you set the channel to, you have to right click on this and change it to forward scatter. Forward scatter tells you the size. This one, right click on it to give you the size scatter. Then you'll see the 2 calibration beads that you used. Okay, so this is the 2 micron beads and this is the 3 micron beads. So if you drag here, it's bigger. Okay, so basically it will tell you the size so that when you look at your sample, it'll be easier to know where your cyanobacteria is. So if you remember during our lectures, Chinchococcus, if you look under the epifluorescence microscope, you can do a direct count. It will fluoresce orange. But for Prochlorococcus, you cannot. You cannot use epifluorescence microscopy. You have to use flowcytometry. Okay. The main problem is because the fluorescence fades very fast, so you need a flow cytometer in order to visualize them. So next we have to create region. So you right click and you say create new region. So you have to click here. Okay, so that will be your R1 population. It's red in color. You right click here. You can rename it. Let's rename it as 2 micron. Okay. So next we'll create another region here to indicate to indicate the 3 micron region. Okay, so now we have the dot plot for this. Okay, so if you want to see, visualize the histogram, what you can do is you can actually just click here at the red part and pull into this. Okay, so when you do that, let's change this into your forward scatter also. Then you can see your 2 beats here, 2 micron and 3 micron beats. Okay, so this is your standard beats, your size calibration beats. Okay, so now let's look at one sample. Let's look at the dot plot again, a new one. Now we open up a sample. Oh, okay, my computer is a bit too slow. So again, forward scatter for size and granularity. Okay, see, so this is the problem when you're working with environmental samples. You have a lot of noise. I have a lot of debris that is unknown. Okay, not clear. So the mistake is with us because we did not pre-filter the sample. Probably if you pre-filter it by 2 micron, then you only get your picoplankton fraction, then you won't have so many problems. But then you can see that this is the 2 micron fraction, this is the 3 microns fraction. So according to the forward scatter here, so anything less than this will be your picoplankton. Okay, but now you want to look at your color. So this is your, so let's make another one. What we want to do is let's do the size here. But here we change it to the Veo Green A. Okay, so Veo Green A will give you, Veo Green A is for Shinicococcus. Okay, so Veo Green A, the channel here will give you the Shinicococcus fluorescens. So all these channels have their spectral wavelengths. So you have to look at the wavelengths and see which one is suitable for the organism that you're looking for, or for the organism that you're looking for, or for the fluorochrome that you have used to stain your organism with. Okay, so for Prochlorococcus, it's APC Veo 770A. Okay, so let's make another one. Okay. So this is again for scatter for the size. APC view 770A. Okay. So you can see the difference immediately. You have a lot more. Signal for the cyanobacteria, for the Shinecococcus, relative to the Prochlorococcus. This is the red part, so this is 2 micron and below. Okay, so this is most the quadrant here are all noise. Okay, so if you were looking for the population, so what we'll do is you have to again create region. Okay, this is the 2 micron beads, so we'll collect from 2 micron onwards, but to avoid the noise at the bottom, so what we do is we'll probably just click here. Okay, so this is the, you rename it as your Shinecococcus. Okay, then for here you create a region here. For the red, if you can see clearly, you can see some of the red signal here, so this will indicate the 2 micron. Okay. So you can rename it as Prochlorococcus. Okay, so you have the two populations here. So if you want statistics, you can, like, if you want to know how many, what you can do is the calculation tool, statistics. Okay. Oh, not this one. Oh, okay. It's here. So from the graph itself, you just create your statistics. Okay, so the events are all here. The population is here. So here, same. Okay, so you have all this here. Okay, you can see the Shinecococcus events. So this is the count, and this is the count for Prochlorococcus. Okay, of course, usually we have to calibrate it. We calibrate by using. We usually calibrate only the Shinecococcus. Because Shinecococcus, we can also use epifluorescence microscopy to count. So what we'll do is we'll run flowcytometry together with the epifluorescence direct counting, and then we'll compare the two data. So it should be one-to-one relationship. Okay. But usually the flow cyto will give a little bit slightly higher counts because